Good morning. So I would like to discuss something a little different, not a trial, these two really nice randomized trials. This is an observational cohort study. This is the Pathfinder study, which looked at a prospective cohort using a multi-cancer early detection blood test. These are my disclosures, but I have no commercial interests, no compensation from the sponsor of this blood test, which is, the study is sponsored by Grail, which is a subsidiary of Illumina. So let me tell you about the study participants. This was a study to investigate a whole new paradigm, a whole new approach to how we screen for cancer. So I think, as you know, cancer screening in older adults is not one-stop shopping, but we have different tests that we undergo for each distinct type of cancer. This study looked at adults who were 50 and older, and they were eligible to participate in this study if they belonged in one of two cohorts. First was the high-risk group, and these were people who had a previous history of cancer but had not had any disease in at least 3 years. So if they had a cancer in the past but had been free of any treatment, any disease for at least 3 years, if they had a history of smoking, and that was simply, in your life, have you smoked more than 100 cigarettes? Or if they were at genetic or familial risk of cancer. So that was the high-risk group. And the other group simply did not meet any of these criteria. So otherwise healthy adults. I mean, they could have had other medical problems, but they didn't have the conditions on the left. So this is how the study worked. We offered a blood test to 6,600 of these individuals. Around 2 weeks later, the results came back, the results came to the physician, and the physician gave the patient the result. For the vast majority of the patients, 6,529, they received a negative test result, which simply said, this blood test found no evidence of cancer in your blood. And thank you very much for participating. We'll check in with you again at 12 months to see if any cancer was diagnosed. That was it. That was the end. Please continue all your regular cancer screening. Do exactly what your doctor says. That was the extent of participation for the 6,529. However, the individuals who received a positive test, so the signal on the test was positive, 92 individuals. The test result included that the test was positive, that there might be a cancer, and also a prediction of what type of cancer it might be. And this was a clue to help direct physicians as to where to look, where to pursue the workup. Workups then were undertaken. And for 35 individuals, a cancer was identified. And in 57 individuals, cancer was not identified as a part of these workups. And these individuals were also followed for a year for the end of study assessment to see a year later whether any cancers were identified. So, to be clear, this meant that the blood test identified cancer in 35 people, or 1.4%. Interestingly, these 35 people were found to have 36 cancers. We had one subject who had both a breast and an endometrial cancer. So 35 people, 36 cancers, or 1.4% of everyone who had these blood tests. As you know, we divide cancers. We think of them as solid tumors and hematologic malignancies that arise in the blood system. 19 of the tumors were solid tumors, and 17 were blood malignancies, and the little map shows you the specific types of cancers. I think what's exciting about this new paradigm and concept is that many of these were cancers for which we do not have any standard screening. So not breast, not colorectal cancer. So just by the numbers, 24 of the 35 cancers occurred in individuals who fell into the high-risk group cohort. 11 were not. 7 of the cancers were found in people with a history of cancer. 14 of the 35 cancers were diagnosed at early stage, and 26 of the 35 are in cancer types for which there is no standard screening, and standard screening was defined based on standards set by the United States Preventative Services Task Force, and it's near identical to the European criteria. So how well did this blood test perform? So 1.4% were positive. The specificity, which is a concept we think about in population health that means what was the probability that somebody without cancer received a negative blood test, was, I need my glasses, I can't see on the slide, 99.1%. The positive predictive value, which is a probability that a person with positive blood test has cancer, was 38%. And the number of people that this meant that we would need to screen to find one cancer in this test was 189. Those are the results. The cancer was also very good at predicting the, what we call the tumor type of origin. So obviously there's immense interest in continuing to refine this assay and to have the assay get better and better. And the concern is always the false positives, that the test is picking up some people and saying that they have cancer when they really don't. So the company that makes the assay is continually working on refining it. So they specified in advance that they were going to apply a different version of this test, a refined version of the test, and they reanalyzed all the specimens, all the 6,600 specimens with a refined blood test. And I've shown you here the performance of the refined blood test. So importantly, it identified fewer patients as having positive signals, so only 0.9%, not 1.4%. But the specificity improved to 99.5, as did the positive predictive value at 43%. Of course, this also means that you need to screen more people to identify one positive cancer, so not 189 any longer, but up to 263. And the ability to predict the origin of the cancer was quite similar. And we just heard a nice presentation about head and neck cancer. I myself had a head and neck cancer patient identified. When we got this test back, it simply said signal positive or negative. The signal said positive, but then the report also said possibly head and neck cancer. Of course, then one starts with a visit to an ENT and a careful oral exam, and one could start there. So the signal of origin was very, very helpful in directing the type of workup.
Excuse me.
Yeah.
We are running out of time.
So the other key points where we looked at how long it took to get these tests results, it took under 3 months for the most part to get the workups completed. So the overall impact is that MCED screening was safely implemented in adults over 50. When the blood test was positive, the workups were typically done in less than 3 months. The blood test typically predicted the origin of the cancer. And I think this is really a new concept. It shows that it is feasible to detect cancers using blood tests. The study was not designed to determine whether this technology can decrease cancer mortality, which is obviously the purpose of cancer screening. It's premature for that. And I think it's key to emphasize that as this technology develops, people must continue with their standard cancer screening. But this still suggests a glimpse of what the future may hold with a really very different approach to cancer screening. So that's it.